Hello friends, welcome to my channel Pathology Master. Myself Dr. Parth Goswami and I am the consultant pathologist. Hope this video will find you in a good health. So now we will understand a very important topic from neoplasia, that is the P53. It is also known by the name guardian of genome because it provides the genomic stability. That's why it is also known by the another name, that is molecular policeman, right? That is the another name of P53. Now name itself suggests this P53 is having the function of cancer prevention. That's why this topic is very important. So as per the new curriculum, any topic should be covered with the learning objectives. So which are our learning objectives? First we will understand what is P53. Then we will understand the cell cycle activation, how the particular cell cycle movement happens. Then we will understand which are the checkpoints for the DNA repair. And once you understand these two topics, you can easily understand the function of P53, and so that you can understand what happens if P53 is mutated. If P53 is mutated, then it will lead to cancer formation. So guys, first of all, you need to understand that P53 is one of the tumor suppressor gene in our body. What do you mean by tumor suppressor gene? They are the genes that prevent the cancer formation. They prevent the cancer formation. This TP53 will produces the P53 protein that is responsible for cancer, that is responsible for prevention of the cancer. Here the P stands for protein and 53 stands for weight of protein, that is the 53 kilodalton, right? The location of the P53 is over the chromosome number 17 short arm, right? And it is present in all normal tissues, right? Okay, now we will see how the particular cell cycle activation will happen. So guys, you are very well familiar that a cell will undergo cell cycle movement from the G1 phase to M stage, right? G1 is pre-synthetic phase, then the cell will enter into synthetic phase, then it will enter into the post-synthetic phase, and finally it will undergo mitosis. So this movement from G1 to M is known by the name cell cycle, right? Now we will understand how the cell will move from G1 to M stage, right? There are certain steps which leads to cell cycle movement. So we will understand how it happens. So guys, first of all, the movement from G1 to S is not an easy, right? It happens because of one important gene, that is RB gene. We will see how it happens. Usually guys, RB gene is in the active form is hypophosphorylated. RB gene is also known by the name retinoblastoma gene. Full name is retinoblastoma gene. In the active state it is hypophosphorylated. It will bind and inhibit the E2F gene. This RB gene will inhibit your E2F gene, that is responsible for transcription, right? So it will inhibit this E2F, so no transcription will happen and there will be no cell cycle movement normally. But when there is a growth signal, when there is a growth factor signal, when the cell have to move into the cell cycle, what happen? This RB gene is become inactivated, right? Normally it is activated, but when the cell cycle movement needed, when there is a growth factor signal, right, this RB gene is inactivated, and once it get inactivated it will hyperphosphorylate, and because of the hyperphosphorylation of RB gene, this particular complex is getting breaked, right? The breakdown of RB plus E2F complex will happen, and so your E2F will get released, right? Your E2F will get released and will be activated, and once it get activated it will induces transcription of many substances, that is the cyclin E, cyclin A, DNA polymerase, etc. And so the cell will move from the G1 to S phase, right? So we can say that RB gene is a gatekeeper of your gene, that is responsible for movement of cell from the G1 to S. So in this way the cell will move in its first step from the G1 to S, and this particular movement will be helped by one particular substance that is produced during the cell cycle, that is known by the name cyclin. you have to remember the cyclin is produced or synthesized during the cell cycle, right? And the sequence of formation of the cyclin is D, E, A and B, means the cyclin D and E will act at a G1 to S transition. So the movement from G1 to S is carried out by the cyclin D and cyclin E, right? And this particular cyclin will activate the cyclin dependent kinase as well. It will activate particular cyclin dependent kinase. Cyclin D will activate cyclin dependent kinase 4 and cyclin E will activate the cyclin dependent kinase 2. So these two complex will cause the cell movement from G1 to S, and the movement of cell from G2 to M will be carried out by cyclin A and B, and for particular corresponding cyclin dependent kinase is 2 and 1, right? So guys, this is about the cell cycle activation. Now if there is any DNA damage in your cell, then what happen? The DNA has to be repaired, and the repair of the DNA is happen at the 2 point. First point is G1 to S and second point is G2 to M transition. So this transition period, this two transition period is known by the name checkpoint, right? It is known as an checkpoint. So there are two checkpoints for DNA repair or cell cycle arrest. So these two checkpoints are known by the name G1S checkpoint and another one is G2M checkpoint, right? This is G2M and this one is G1S checkpoint. So this G1S checkpoint will repair the DNA before replication and G2M will repair the DNA after replication. And guys, you have to remember that P53 will act as both these checkpoint. It will activate your both checkpoint, right? It will induces your DNA repair, and if it cannot repair, then it will stop the cell cycle. So this two checkpoint is mediated by P53. Particularly it act as a G1S checkpoint. Now guys, after understanding the cell cycle activation, you can understand the function of P53. Now you can understand how P53 will provide you the genomic stability. So guys, there are basically four main functions of P53. One function is cell cycle regulation. Another function is DNA repair. Third function is to induce apoptosis. And fourth function is to do the cell cycle arrest, either temporary or permanent. Temporary cell cycle arrest is known by the name quiescence, and the permanent cell cycle arrest is known by the name senescence, that will be carried out by microRNA and link RNA. Now I will show you in the figure that how particular P53 is getting activated when you have the DNA damage. So guys, now I will explain you the function in the detail. Then you can understand how cancer happen if P53 is inactivated. So guys, normally P53 in the normal cell, in the normal tissue, it is very unstable, right? It is very unstable and it degrades very rapidly. Why it degrades very rapidly? In no stress condition, in the healthy cell, P53 half-life is only 20 minutes. So you can predict that within 1 hour, the P53 will get degraded. It's very unstable and degrade very rapidly. Why? That is because one particular gene that is known by the name MDM2 gene. It particularly targets the P53 protein. This MDM2 having attraction towards the P53 normally, and it will degrade your P53. It will reduces your P53. So in the normal healthy cell, you don't have increased number of P53. But now we will understand what happened when there is a DNA damage. So guys, this particular figure is prepared by me and it is very easy to understand. So guys, whenever you have the DNA damage, what happened? Your DNA damage sensors will get activated. When there is a DNA damage, the sensors are getting activated, particularly ATM gene, that is ataxia telangiectasia mutated gene, and the ATR gene. These two genes are getting activated. Once they get activated, they will phosphorylate your P53. They will do the phosphorylation of P53, so it will get released from the MDM2, right? It will reduces the attraction of MDM2 towards the P53. So because of the sensor activation, the P53 will be released from MDM2, and so your blood level of P53 will be increased. So your P53 will be increased in number, right? They will increase. And guys, once the P53 increases, it upregulates the one specific protein that is known by the name P21. Now, what is the function of this P21? This P21 is cyclin dependent kinase inhibitor. It will inhibit your CDK, and you can predict that if your CDK are getting inactivated, suppose these CDKs are inactivated, then what happen? Your cell cycle will arrest. So your cell cycle will particularly arrest as G1S phase. The cell cannot move from G1 to S because it is a damaged cell. You need to stop it, and this function is carried out by P53. And once the cell is arrested at this stage, it gives a time for the cell to repair itself. So there will be DNA repair in this time, right? And the repair is also induced by P53. See guys, what is the function of P53? It will induces the gene that is responsible for DNA repair. So P53 will help you in the DNA repair as well. It will induces the GAD-D45, right? This particular gene is having the function of DNA repair. So now it will help in DNA repair, and once the DNA is get repaired, the P53 will degrade because there is no need of P53 after the DNA repair. So it will get degrade, and your repaired cell will further move into the cell cycle. So cell cycle will continue after the repair. But guys, this is not the scenario every time. Sometimes what happen, your DNA cannot repair, right, by the P53. Sometimes DNA will not get repaired. So at that time what happen? Suppose your DNA is not repaired, then the cell will undergo either apoptosis or will undergo senescence, right? The apoptosis is because of activation of Bach's gene, that is the pro-apoptotic protein, or Puma gene. These are the pro-apoptotic proteins, right? Or it will undergo senescence because DNA cannot get repaired. The senescence means permanent cell cycle arrest that is carried out by microRNA. So guys, now you can understand that what the P53 will do into your body, right? If you have the DNA damage, it will try to repair your DNA, right? If your DNA cannot repair, first of all they will try to repair it, and they will try to save the cell. But if DNA cannot repair, then what they will do? They will stop your cell cycle. They will not allow the mutated cell or damaged cell to enter into the cell cycle, right? They will arrest the cell cycle to prevent the cancer formation. Or they can do the apoptosis of the damaged cell. They will remove the cell through the process of apoptosis. So we can say that guys, P53 is having the central role in maintaining the integrity of the genome. They will maintain the integrity of your genome, means they will not allow the damaged cell to enter into the cell cycle, and thus they will protect your genome, right? And that's why it is given one of the beautiful name, that is guardian of genome, and that's why it will prevent the cancer formation. So it has been observed that when you have the mutated P53, more than 70% patient of the cancer have show the mutated P53 gene, because when you have the mutated P53 gene, you will have more chance for cancer formation. So it has been observed that in greater than 70% of the human cancer, the P53 is mutated, and particularly it can lead to all type of cancer, but particularly it will lead to formation of lung cancer, colon cancer, and the breast cancer. That's why P53 is very important for your body, right? In some cancer, what happened, like that of sarcoma. In the sarcoma, your MDM2 is overexpressed. So what happened? they will reduces your P53, and because of reduction of P53 your genome is not protected, and so you will have the cancer. So that is also not good for your body if MDM2 is overexpressed like that in case of an sarcoma, right? If you have the P53 mutation from the birth, right, if you have the mutant P53LA, then it is known by the name Liefeld syndrome, and it puts you at the risk for cancer formation, right? So this syndrome is very dangerous for your body. You can develop cancer earlier in your life if you have the mutated P53. This is the same diagram showing you the function of P53, right? It will, if there is a DNA damage, then it will try to repair it through get D45. If the repair fail, then it will induces the apoptosis or it will lead to senescence, right? If the DNA repair cannot happen, then it will lead to apoptosis or cellular senescence. And by the induction of P21, that is cyclin dependent kinase inhibitor, it can arrest your cell cycle at the G1S phase. So guys, now you can understand what happen if P53 is inactivated, right? What happen? Suppose your DNA is damaged, right? Your DNA is damaged. Now if you don't have proper functioning of the P53, then what happen? There will be no damaged cell cycle arrest at G1S phase, right? P53 will not function. So what happen? This damaged cell will not be stopped and they will undergo cell cycle movement. So there will be no cell cycle arrest if your P53 is inactivated. Additionally, they will not do any DNA repair because your P53 is mutated. There will not be any apoptosis of the damaged cell. So the cell progress with the damaged DNA, right? The cell progress with the damaged DNA, and this will put you at the risk for cancer formation. So if your P53 is inactivated, then you can develop the cancer earlier in your life. So guys, again I am telling you the function of P53. Just remember this master chart. It is a very important master chart prepared by me. If there is a DNA damage, then your DNA damage sensor will get activated like that of ATM genes. It will phosphorylate your P53. So it will be released from MDM2. Normally MDM2 inhibit the P53. So it will get released from MDM2, and so your P53 level will be increased. Increased P53 will activate your P21. This P21 is cyclin dependent kinase inhibitor. So you can predict that if your CDK inhibitors are activated, then what happen? Your cell cycle will arrest. So cell will arrest at G1S phase. So it will allow the time to the cell for the DNA repair. Additionally, P53 induces the get D45 that lead to DNA repair. Once the DNA is repaired, P53 degrade and the normal cell cycle movement will happen, right? But if DNA is not repaired anyhow by the P53, then what P53 will do? Then they will lead to apoptosis through the activation of Bach's gene, or they will lead to cellular senescence, means permanent cell cycle arrest through microRNA stimulation. They will lead to cellular senescence, that is permanent cell cycle arrest. So this is the function of P53, and that's why it is known by the name guardian of genome. And if it is mutated, then it can lead to cancer formation. Now we will see some of the one-liner MCQs. Usually the case type of MCQ is not being asked from the P53 topic. So which gene is mutated in Liefeld syndrome? Obviously your answer will be P53. Okay. Second MCQ, which gene is responsible for instability and reduced lifespan of P53 in the normal cell? Obviously, why P53 half-life is very short, like that of 20 minutes in the normal cell? That is because it is unstable and degraded by the one gene, that is MDM2. It attracted toward P53 and it will damage the P53. That's why instability and reduced lifespan of P53 is because of MDM2 gene. So your answer will be MDM2. Okay. P53 upregulate the which gene that causes the cell cycle arrest when DNA damage happen? Obviously the cell cycle arrest is because of activation of P21 by the P53. So your answer will be P21. 1. Okay, thank you very much guys. So guys, kindly subscribe my channel and press the bell icon so that you can get notified whenever I am posting the new video, right? If you have any question in your mind, then you can ask in the comment section. I will surely answer it. Thank you very much guys, and see you soon in the next video. Thanks. Take care.